Good day. So last meeting we ended with the different members of Enterobacteriaceae, which is of course included in the gram-negative bacilli group. Okay, so for now, let us continue with the other gram-negative bacilli. So let us start first with non-Enterobacteriaceae glucose fermenters. Okay, so these three organisms, they are also enterics. However, they do not belong in the Enterobacteriaceae family. Okay, so let us first start with Vibrio cholera. So of course, it is the causative agent of cholera, okay, because of its toxin, okay, the choleragen. And because of that toxin, okay, the manifestation of cholera is diarrhea, no, which is characterized by having rice watery stool, okay? Vibrio cholera is non-halophilic in contrast with other members of Vibrio Naceae. When you say halophilic, of course, the organism requires high salt concentration. However, not all of them are halophilic, and one of which, of course, is Vibrio cholera. Now, the other two, just to mention, we have Vibrio parahemolyticus and Vibrio vulnificus. Later on, I will draw a table for us to differentiate Vibrio cholera with the other two. So, Vibrio parahemolyticus and Vibrio vulnificus, those are Vibrio species that are halophilic. Vibrio cholera is motile and it is positive for the string test. The string test makes use of 0.5 sodium deoxycholate. And for the culture, we could use alkaline peptone water, and we could make use of TCBS, wherein your Vibrio cholera will give out or will grow yellow colonies because Vibrio cholera is a sucrose fermenter. Okay, so if you are a sucrose fermenter, you will have yellow colonies in TCBS, while for non-sucrose fermenters, they tend to have green colonies. Okay, so going back no, to the table I mentioned, here's a quick differentiation, okay, between Vibrio cholera, Vibrio parahemolyticus, and Vibrio vulnificus. Okay, so first, let's differentiate them based on their growth on TCBS. Okay, TCBS is thiosulfate citrate bile salt agar. No, thiosulfate citrate bile salt sucrose agar. Okay, so based on their growth on TCBS, your Vibrio cholera is positive for sucrose fermentation, thus nagkakaroon siya ng yellow colonies, whereas the other two are sucrose non-fermenters. So if non-fermenter ka, green yung magiging kulay ng colony mo. Okay? Next is we could also differentiate them based on their sensitivity on O129. Okay? And resistant no yung ating Vibrio parahemolyticus, while Vibrio cholera and vulnificus are sensitive. Then based on their need for a high salt concentration, your Vibrio cholera is non-halophilic, your hemolyticus is halophilic, and your vulnificus is also halophilic. Actually, your vulnificus is strictly halophilic. Okay? So, usually yung ating Vibrio, they are associated of course with oyster-related injuries. Specifically, no, yung ating Vibrio vulnificus. Okay? Oyster-related injuries. So, pwede no, it could be a gastrointestinal manifestation because of the consumption of raw oyster, or it could be a wound infection because nasugatan ka dun sa shell ng oyster. Okay? Now, let's proceed to Campylobacter, Helicobacter first. Helicobacter causes peptic and duodenal ulcers. It is a microaerophil, okay, which means it requires or it grows or thrive at a low oxygen environment. Urease, oxidase, and catalase positive. It is motile with 4 to 6 flagella. And for the identification, we could make use of staining of tissues. Pwede din yung culture biopsies obtained of course from the stomach or duodenum, considering the manifestation of Helicobacter pylori. Okay? So usually there are a series of tests, no, or there are choices, no, of tests for the diagnosis and for us to know if whether Helicobacter pylori is already eradicated from the body of the patient. Okay? And what are they? So pwede tayong gumamit, no, for diagnosis. Okay? Test for diagnosis and eradication. Okay? To know if the organism is already eradicated. So of course we have the urea breath test. Okay? Considering that Helicobacter pylori is a strong urease positive organism, then pwede ding gamitin yung stool, okay, stool antigen test. And yun nga, no, yung ating histologic examination of stomach or duodenal biopsies. Okay? And lastly, for this group, we have Campylobacter jejuni. Okay, so Campylobacter jejuni also is a microaerophilic organism, and at the same time, it is also capnophilic. And alam naman natin, no, pag sinabi natin capnophilic, it requires, no, a higher concentration of carbon dioxide, no. Specifically, no, ano ba yung tamang timpla, no, ng environment when it comes to the growth of Campylobacter jejuni. So take note of this. Usually, no, nagre-require siya ng 5% oxygen, 85%, okay, 85% nitrogen gas, and 10% carbon dioxide. Okay, so this is the mix, no, or the specifications, no, for the environment optimum, no, for the growth of Campylobacter jejuni. So this is also one of the causative agents of gastroenteritis because this is an enteric pathogen and is also associated with Guillain-Barré syndrome. It is oxidase and catalase positive, similar to Helicobacter pylori. However, it is also heparite hydrolysis positive. Guys, I want you to take note of the positivity of Campylobacter jejuni sa heparite hydrolysis test because this is one of the keys for us to identify Campylobacter jejuni from other Campylobacter species because sa lang yung nagbibigay ng heparite hydrolysis positive result. Okay? It has a darting motility and it grows at 42 degrees Celsius to 45 degrees Celsius. Okay? However, no, i-specify ko lang, no, si Campylobacter fetus. Okay? Because Campylobacter fetus grows. Okay? It grows at 37 degrees Celsius. Okay? So, usually, no, if we are to differentiate Helicobacter versus Campylobacter, because meron silang similarities. Both of them are microaerophilic, both of them are oxidase and catalase positive. However, if we are really to differentiate them, take note that Helicobacter pylori will give a positive result in the urease test. Na negative naman dun yung Campylobacter. And Campylobacter grows at 42 to 45 degrees Celsius, whereas Helicobacter doesn't. Okay? So, that would be all for our non-Enterobacteriaceae glucose fermenters. Okay? So, now let us proceed to gram-negative non-fermentative bacillus. Okay? So, marami sila, no, but we are to place emphasis on Pseudomonas aeruginosa and Burkholderia species. So first, we have Pseudomonas aeruginosa. Okay, this is a common causative agent of nosocomial infections. When you say nosocomial, those that are acquired, no, usually sa mga healthcare settings. Okay, so this is the agent of blue or green pus owing to its ability to produce a certain pigment. It is an obligate aerobe organism, oxidase positive and motile. It grows well at 37 degrees Celsius and take note of this, 42 degrees Celsius. Sa katunayan, the growth of Pseudomonas aeruginosa at 42 degrees Celsius is one of the ways in order for us to differentiate this organism from other fluorescent Pseudomonas species or from other Pseudomonas species that are also capable of producing pigments. Okay? Recall question kasi ito, no? Yung growth niya at 42 degrees Celsius is is a way, no, to differentiate Pseudomonas aeruginosa from other, okay, pigment producing Pseudomonas species. Now, isa no sa mga clue natin for us to know that it is Pseudomonas aeruginosa na nag-grow is because of its characteristic grape-like odor, or minsan tinatawag din siya na corn tortilla odor. Okay? This is because of the substance amino acetophenone. Okay, amino acetophenone. Then of course, it has a blue and green pigment. That's why nga tinawag siya na agent of blue or green pus. So the blue pigment is pyocyanin, whereas the green pigment is pyoverdine. Okay? Actually, yung pyoverdine, it could be green or yellow-green, no, yung kanyang pigment. So this is a most common cause of respiratory infections among cystic fibrosis patients. And usually patients in the ICU who are mechanically ventilated may quickly become colonized with Pseudomonas aeruginosa. The reason why I mentioned about the nosocomial infections. Another thing, okay, other than the pigments that I already have mentioned, no, the pyocyanin and pyoverdine, we also have other two pigments that Pseudomonas aeruginosa is also capable of producing. This includes your pyorubin, okay, which is of course red, and we have pyomelanin, okay, which is a brown pigment. Okay? Another thing, talking about pigmentation, of course, if we want to enhance, no, if we want to enhance the pigmentation of Pseudomonas aeruginosa, we could culture it in cetrimide agar. Okay? Cetrimide agar, no? So this is an agar that enhances, okay, pigment production. Okay? So that would be all for Pseudomonas aeruginosa. Let us proceed to Burkholderia species. So dito meron tayong tatlo. First, we have Burkholderia malay, a non-motile pseudomonad, okay, which causes glanders disease. Okay, glanders disease. So this is a disease of horses and similar animals that can be transmitted to humans. No, so it is a zoonotic infection. Okay. Then we also have Burkholderia pseudomalae, which causes melioidosis, or an endemic glanders-like disease of animals and humans, which is also referred to as the Whitmore's disease or Nightcliff Gardiner's disease. Okay, so your Burkholderia pseudomalae is one of is a potential, okay, bioterrorism agent, no? That's why tinawag nga siyang Vietnamese time bomb, okay, because it is a potential, okay, bioterrorism agent. Okay? So another thing, no, that I would like you to take note about Burkholderia pseudomalae, take note of its wrinkled colonies. Okay, it's wrinkled colonies on sheep's blood agar plate. Okay? And lastly, we have Burkholderia cepacia. Okay, a common recall question in the ASCP exam. Yung parating tanong is yung organism daw associated with patients with cystic fibrosis, no? However, take note that this is just the second most common cause. Kasi yung most common cause of respiratory infections among patients with cystic fibrosis is Pseudomonas aeruginosa. Sa katunayan, guys, take note of the 4 organisms that must be screened always in patients with cystic fibrosis. I do not know if I already have mentioned that in the previous lectures. But again, take note of the 4 organisms. First, we have Pseudomonas aeruginosa, Burkholderia cepacia, Staphylococcus aureus, and Haemophilus influenzae. So those are the 4 organisms that are commonly infecting patients with cystic fibrosis. Thus, cystic fibrosis patients must always be screened sa 4 na organisms na yon. Okay? Another thing, no, natatandaan natin about Burkholderia cepacia is that it is resistant to aminoglycosides. Okay? Now, let us go to the miscellaneous gram-negative non-fermentative bacilli. So, mga ano to, no? Mga kapatid ng Pseudomonas aeruginosa and Burkholderia cepacia. So, I'll just point out those that are that have been asked already, no, in the ASCP exam. Of course, we have Stenotrophomonas maltophilia, okay, which is a nosocomial pathogen, also causing respiratory infections. Okay, so this is a nosocomial. Okay, a nosocomial pathogen. And usually the manifestation is respiratory. Okay, so take note of its lavender green colonies on ship's blood agar plate. Okay, yellow pigment and TSI, and it is a motile organism with Lophotrichus flagella. Okay, hinighlight ko dyan guys yung mga medyo unique, no, na biochemical properties sa kanila. Okay, so it is oxidase negative. Okay, oxidase negative kasi technically yung group na ito, they are expected to be oxidase positive. Katulad ng ating Pseudomonas aeruginosa, no, katulad ng ating Burkholderia species. So they are expected to be oxidase positive. That's why I highlighted those that are oxidase negative. Okay? So it also has an ammonia-like odor and it is resistant to carbonicillin but sensitive to kanamycin. Okay? Another thing pala, tatandaan din natin, no, that Stenotrophomonas maltophilia grows also, no, grows at 42 degrees Celsius. Okay? Next, gusto ko ding i-emphasize, no, si Acinetobacter. Okay? So it has round opaque mucoid colonies on blood agar plate. Okay? Purple colonies on McConkie, and because of its characteristic purple colonies on McConkie, it may be mistaken, no, or it may resemble lactose fermenting organisms. It is non-motile. Okay? Then there are two organisms, no, na kailangan nating i-take note dito, no. We have Acinetobacter baumannii and Acinetobacter lofi. Okay? Take note lang natin guys. Ito yung parang pinaka-difference ng dalawa. Tatandaan natin, no, that Acinetobacter baumannii, okay, is commonly resistant, no? Mga resistant siya sa mga antibiotics, whereas Acinetobacter lofi, okay, is susceptible, okay, to antibiotics. Similar to Stenotrophomonas, it is also oxidase negative. Okay? And take note, it can be mistaken, okay, as Neisseria. Then we have Chrysobacterium meningosepticum. However, no, lumabas ito, no, sa ASCP exam, but the new name already of this organism. So yung bagong pangalan ng organism na ito is Elizabeth, okay, Elizabeth Kinja, okay, meningoseptica. Okay, Elizabeth Kinja meningoseptica. So it is MAC negative because that is common. Okay, that is common among these organisms, no? Mga ano sila, no, mga fastidious, no? So usually they do not grow on McConkie agar. Okay, so hinighlight ko din dyan guys, no, yung mga MAC negative. So this is a filamentous organism. Okay, ibig sabihin meron siyang thread-like structure. Okay, so meron siyang thread-like structure, MAC negative, yellow pigment on sheep's blood agar plate because of flavin, and it is non-motile. Okay, so take note lang natin dito, no, this is associated with the use of nebulizer and also linked to formation of yellow milk. This is an agent of meningitis sa mga neonates. However, meningitis is only a rare condition caused by this organism. Okay, so neonatal meningitis. So rare lang, no? Rare manifestation lang ng organism na ito. Okay? And we are to discuss Echinella and Kingella together with the Hasek group later. Okay, let us now proceed with Chromobacterium violacum. So this one is an opportunistic pathogen. Okay, an opportunistic pathogen. Of course, no, do not forget about its violent pigment. It is motile with polar flagella and also grows at 42 degrees Celsius. Okay, so together with Kingella species, they are capable of fermenting glucose. Okay, they are capable of fermenting glucose. No, so it is also a bit unique about Kingella and Chromobacterium violaceum, considering that technically yung pangalan ng group is gram-negative non-fermentative organism. Okay, that's why being able to ferment glucose is something that is ironic, no, about these species. So we have to take that into consideration, or that's something na pwedeng maging trivial about them, kaya kailangan nating i-take note talaga. Okay? So yan po no yung ating gram-negative non-fermentative organism. Okay? So let us now proceed to the last group, no, for our gram-negative bacilli. We have the fastidious organism. So of course, let us start with the Haemophilus species. Okay? So gusto ko munang tandaan niyo no yung mga characteristic na ito when it comes to Haemophilus species. Okay? They are pleomorphic. Okay? Pleomorphic. They are Cocobacilli. Okay? And they do not grow on sheep's blood agar or Maconchi agar. However, they grow on chocolate agar. So take note, no growth on sheep's blood agar plate or Maconchi. Okay? However, they grow gray colonies on chocolate agar plate. Okay? So take note, they are oxidase and catalase positive. The preferred incubation is at 35 degrees Celsius to 37 degrees Celsius with 5 to 10% carbon dioxide, making them, of course, capnophilic organism. So they can be differentiated based on their X and V factor requirement. So yung X factor, yan yung tinatawag natin na hemin. So usually they are getting that from chocolate agar. Then we have V factor, which is otherwise known as the NAD coenzyme. And that NAD coenzyme can be taken from other organisms. No, that's why since other organisms are providing this V factor, nalalaman natin if whether the organism is requiring V factor if they are exhibiting satellitism around these organisms. So ano yung mga organisms na ito, no? So they are exhibiting satellitism, okay, around S. aureus. Okay? They could also exhibit that around S. pneumoniae. Okay? And around Neisseria. Okay? Again, the satellitism is a clue that the organism is requiring V factor. Okay? So, we have 3 organisms to emphasize here. We have first Haemophilus influenzae, which is otherwise known as the Pfeiffer's bacillus. Okay? So this is the main cause of meningitis in children below 5 years old and also a major cause of epiglottitis. Okay? So another thing no natatandaan natin about this recall question, no? Recall question in my exam, Haemophilus influenzae is the organism that must be tested for beta lactamase production. Okay? Tinanong na yan sa aking exam, no? It requires, no? Requires testing for beta lactamase production. Okay? So Haemophilus influenzae yon. Okay? And there are several types, no, or subtypes of Haemophilus influenzae. So take note lang natin that type B, okay, type B is the cause of the most serious infection. Okay? Next, let us proceed to Haemophilus aegypticus. This is otherwise known as the Kochweek bacillus, Kochweek's bacillus. And of course, this is the agent of pink eye conjunctivitis. Okay? So usually, no, in order for us to be able to cultivate, okay, Haemophilus aegypticus, it requires enriched chocolate agar plate. So usually, that is enriched with 1%, okay, isobitalex. Okay? 1% isobitalex, and it must be held, okay, for 4 days. Okay? To really ensure na kung meron ba talagang Haemophilus aegypticus o wala. And lastly, we have Haemophilus ducreyi, otherwise known as the Ducre's bacillus. Okay? Ducre's bacillus, it causes soft, okay, painful chancre, or tinatawag din siyang chancroid. It has a school of fish arrangement on gram staining, and this is the smallest pathogenic bacteria. Okay? So again, they require 5 to 10% carbon dioxide, okay? And usually, no, for the growth naman of Haemophilus ducreyi, you could incubate it up to 7 days. Okay? At a lower temperature, usually, di ba, 35 to 37, but that is just 32, okay, degrees Celsius when it comes to Haemophilus ducreyi. Okay? Now, as I have mentioned, the Haemophilus species are commonly differentiated based on their X and V factor requirement. So, nakagrupo na yan sila, guys, no? So, usually, yung Haemophilus influenzae natin, Hemolyticus and Aegypticus, sila yung nagre-require ng X and V factor. Okay? Then, si Ducreyi, ito yung nagre-require ng X factor lang, hindi niya kailangan yung V factor. Lahat ng merong para sa kanilang pangalan, sila yung nagre-require lang ng V factor. They do not require X factor. Si Aprophilus, tinatandaan ko dito, wala, no? 'Di ba? Usually, pag narinig natin yung A na term, ibig sabihin absence. So, wala siyang kailangan. Hindi niya kailangan ng X tsaka ng V factor. Guys, another thing, the moment na hindi niya kailangan ng X factor, that makes the organism positive for the D-ALA test. Okay? So lahat ng organism na hindi nangangailangan ng X factor will automatically be positive for the D-ALA test. So the D-ALA test is otherwise known as the porphyrin test. Okay? This is otherwise known as the porphyrin test, which of course detects the ability of the organism to convert ALA to porphyrins. Okay? So detects the ability of the organism, okay, to convert, okay, ALA, aminolevulinic acid, to porphyrins. Okay? So take note, if yung organism nagre-require ng X factor, ibig sabihin ALA negative siya. But if hindi siya nagre-require ng X factor, it is ALA positive. Okay? Now, let us proceed to Bordetella. Bordetella, we have here 3 organisms na i-differentiate natin. We have Bordetella pertussis, parapertussis, and bronchiseptica. So, Bordetella pertussis is otherwise known as the Bordet-Gengo bacillus. Okay? Causative agent, of course, of whooping cough or pertussis, which has 3 stages, the catarrhal, paroxysmal, and convalescent stage. Okay? So, important characteristics of Bordetella, it requires cysteine and methionine for growth. It is an encapsulated obligate aerobe. That's why pwede natin silang masubtype based on its capsular antigen. Okay? It grows sa Reagan low and Bordet-Gengo medium. So, take note lang, pag itinanim siya sa Reagan low, it has a white mother of pearl opalescence, whereas pag Bordet-Gengo, the colonies are resembling mercury droplets. Okay? So, another thing na tatandaan natin dito, no, another characteristic is that Bordetella pertussis is very sensitive to drying. Okay? Also, tatandaan din natin yung kanyang specimen of choice. Okay? The specimen of choice is of course nasopharyngeal swab. Okay, the nasopharyngeal swab. Okay, next, let's proceed to Bordetella parapertussis. It causes pertussis-like syndrome. And lastly, Bordetella bronchiceptica, no, which is the causative agent of kennel's cough, which is a disease of canines. And pag humans naman, it causes chronic respiratory tract infection. Okay, so that would be all for Bordetella. Next, we have Brucella. Okay, so Brucella, traditionally, Brucella is a causative agent of an occupational disease among livestock handlers. Okay? However, today, yung common na manifestation ng Brucella infection is parang uh, foodborne, no? Foodborne infection na because of usually unpasteurized dairy products. Okay? So ulitin ko traditionally, Brucella is a causative agent of occupational disease among livestock handlers. No, that was before. However, today the common manifestation or the common Brucella infection is a foodborne infection after consumption of unpasteurized dairy products. Okay? So Brucella is a normal flora of the urinary and the gastrointestinal tract of different animals, no? So usually nakadepende din sa animal kung anong species siya, no? So for example, we have Brucella militensis, no? Usually seen in sheep and goat, Brucella abortus in cattle, Brucella suis in pigs, and Brucella canis in dogs. Okay? So brucellosis, undulant fever, which is otherwise known as Malta fever or Mediterranean fever or Gibraltar fever or Cyprus fever. No? So this is the condition that is caused by Brucella, and it is also associated with animal abortion. Now, talking about the disease, no, that is caused by Brucella, take note natin guys yung manifestation or kumbaga yung pinaka-distinct na feature ng sakit na kinokos ng Brucella. Okay? So, the disease, okay, is usually characterized by fever, okay? Fever with splenomegaly. Okay? Fever with lymphadenopathy. Fever with lymphadenopathy, hepatosplenomegaly, and malodorous perspiration. Okay? So that is the hallmark, kumbaga, no? That is the hallmark of undulant fever or brucellosis. And take note natin yung specimen of choice. Okay, specimen of choice natin for the isolation of Brucella. Okay, that would be bone marrow. Okay, now important characteristic, Brucella is a gram-negative fastidious bacilli. Okay, obligate intracellular, and it has no capsule unlike Bordetella pertussis. It's aerobic and non-motile. Okay, and it can be grown on Castaneda medium, which is a biphasic medium. Okay. Next, we have Legionella. Legionella is naturally found in both artificial and natural water sources, such as ponds, creeks, and streams, wet soil, water cooling towers for air conditioning and heating systems, shower heads, and plumbing systems. So it can be cultured on buffered charcoal yeast extract agar or Phili Gorman agar. Okay, so take note na itong BCYE yan, recall question na yan, no? Agar for Legionella. So for this stain, we have silver impregnation. Okay? Because usually yung Legionella natin is gram invisible in primary stain smears, no? So that's why the staining method of choice is silver impregnation. Okay? We also have the urine antigen detection test for rapid identification or rapid detection of Legionella antigen, no, in urine specimens, no? So recall question din 'yan. Ano daw yung organism na nadidetect sa urine antigen detection test? Okay? And we also have direct fluorescent antibody test. Okay, this is also a rapid laboratory procedure for the detection of the more common species of Legionella found in clinical samples from the lower respiratory tract. Okay, so we are making use of a fluorescent dye here, no, which is fluorescein isothiocyanate. Okay, and the organism will appear as bright yellow to green, short or cocoa bacilli with intense peripheral staining, no, after doing the DFA test. Next, we have Pastorella. Okay, so to be specific, we have here Pastorella multocida, which is the causative agent of a zoonotic infection or wound infection, usually coming from the bites, no, of usually, no, cats and cat bite. Okay, or pwede din, no, galing sa ibang mga animal bites din. Okay, pero yung pinaka-associated talaga dito is yung cat bite. Kaya nga siya tinawag na causative agent of cat bite fever. Okay? So this is the causative agent, of course, of pasteurellosis, the sheeping fever in cattle's, wound infection, which is most common in humans. So it grows on blood agar plate with a characteristic musty odor, but not on McConkey agar. Okay? It is positive for oxidase, catalase, indole, nitrate reduction, and glucose utilization. And pakidagdag lang, it is susceptible. Okay? It is susceptible to penicillin. Okay? So take note of the cat bite fever. Next, we have Francisella tularensis. Okay? So, causative agent of tularemia. Okay? Or tinatawag din ito, guys, na rabbit fever. Okay? This is also referred to as the rabbit fever or deer fly. Okay? Deer fly fever. Okay? So this is a faintly gram-negative coccobacilli, non-motile and obligate aerobe, and it requires cysteine and cysteine, no? Sorry, the other spelling should be this, no? Cysteine and cysteine for growth, no? Cysteine and cysteine, these are sulfhydryl. Kasi baka gamitin sa exam, sino daw yung nagre-require ng sulfhydryl compounds for growth? So that would be your Francisella tularensis. Okay? So for the culture, no, it can be grown on blood cysteine glucose agar, and it can also be grown on BCYE, no, or the buffered charcoal yeast extract agar, which is also the same agar na pwede nating gamitin for the growth of Legionella. Okay? So pwede din siyang i-grow sa BCYE. Okay? Now, another thing na tatandaan natin about Francisella, okay, is that it is a BSL-3, okay, organism. So of course, no, for the handling of this organism, you have to do it under a biosafety level 3 hood. Okay? Now, also I would like you to take note about the PCR techniques, okay? Because that is the most widely used method, no, for the isolation of for the detection of Francisella. Okay? Because technically, no, yung direct fluorescent antibody staining, it could be useful. However, no, the most widely used method is PCR. So take note, PCR is the preferred, kumbaga, no, this is the preferred method, okay, for the detection of Francisella. So it could this organism may grow on blood agar plate, okay, and in chocolate agar plate. However, the growth is not that abundant, no, and usually it requires a longer incubation period of approximately 36 to 48 hours. Okay, so take note na lang din natin poor growth, no, poor growth, okay, on blood agar plate and on chocolate agar plate. So usually, in order for them to grow, you need to incubate them for around 36 to 48 hours. Okay? Again, emphasize ko itong PCR because that is a recall question. Ano daw yung preferred method for the diagnosis of Francisella tularensis? So take note, that would be PCR. Okay? So pwede din yung iba, no? Pwede din yung direct fluorescent antibody. It is also useful. However, the most widely used method because it is preferred is polymerase chain reaction. Okay? Next, we have, and last, no, for our gram-negative bacilli, we have the HASEC group. So the term HASEC, okay, stands for the members, no, or the 5 members of this group, no. So they are small gram-negative coccobacilli, which are part, no, of the normal oral flora, and they are commonly associated with bacterial endocarditis. So because they are part of the normal flora and they may cause infection, they are classified as opportunistic infection, no? And they may enter the bloodstream, settle on damaged heart valves, and cause a relatively slowly progressive indolent form of endocarditis. So they typically require an additional 1 to 2 days before they are isolated from blood cultures, and they are uniformly susceptible to many antimicrobial agents. Okay? So, sino-sino ba yung mga membro, no, ng HASEC group? So, H here stands for Haemophilus aprophilus. Okay? Haemophilus aprophilus. Okay? The A here stands for Actinobacillus. Okay? Actinobacillus actinomycetem comitans. Okay? Then we have Cardiobacterium hominis. Okay, Cardiobacterium hominis. Then we have Echinella. We have Echinella corrodens. And lastly, we have Kinjela kinjay. Okay? So take note natin yung catalase, lahat sila catalase negative except for Actinobacillus actinomycetem comitans, and all of them are oxidase positive also except for Actinobacillus actinomycetem comitans. Okay? So for the features, no, yung mga most notable features ng mga organisms na ito, kanina we already have mentioned about Haemophilus aprophilus that it has no X and V factor requirement. Okay? It has no X and V factor requirement. And again, since wala siyang X factor requirement, automatically Haemophilus aprophilus is positive for the ALA or the porphyrin test. Actinobacillus actinomycetem comitans, it has characteristic star shape. Okay? Star-shaped colonies. Okay? Cardiobacterium hominis, take note natin, it is endo-positive. And Echinella corrodens and Kinjela kinjay, both of them pits the agar. Okay? Both of them pits the agar. However, tatandaan din natin yung pagkakaroon ng bleach-like odor. Okay? Ng Echinella corrodens, and take note natin yung twitching. Okay? Twitching motility of Kinjela kinjay. Okay? So take note, Echinella corrodens is the causative agent of human bite fever. Okay? Causative agent siya ng human bite fever si Echinella corrodens. Now, another recall question, no, about the HASEC group. Sa ano na daw ba yung bagong pangalan, no, ng certain members of the HASEC? So take note, guys, that Haemophilus and Actinobacillus are now grouped as Aggregatibacter. Okay? Aggregatibacter. So today, it's already Aggregatibacter aprophilus and Aggregatibacter actinomycetem comitans. No? So dalawa lang naman sila yung na-regroup na into Aggregatibacter. Okay? Okay, so let us now proceed sa ating mga anaerobes. Okay, so take note natin for anaerobic organism, ano yung ating specimen of choice? Okay? Specimen of choice natin, of course, okay, aspirate. Yan. Okay? So, ginawan ko na lang ng table, no? So, first we have the anaerobic cocci. So, meron tayong gram positive and gram negative, of course. So, yung unang tatlo, they are all gram positive, then gram negative si Veillonella. For vancomycin, of course, sensitive yung mga gram positive. Okay? Resistant naman yung gram negative, which is Veillonella. For kanamycin, okay, variable yung Phenigoldia magna, no? So it could be sensitive, it could be resistant, okay? Peptostreptococcus is resistant for kanamycin, susceptible yung Peptonephelus and Veillonella. Same, baliktad, no? If ito lang tatandaan natin, if sensitive yung mga gram positive sa vancomycin, resistant naman yan sila sa colistin. Okay? Tapos sensitive naman yung yung gram negative. Okay? Catalase negative sila, variable however yung Veillonella, and SPS susceptibility negative sila except for Peptostreptococcus anaerobius, no? So anong ibig sabihin nitong positivity na ito? So this is susceptible, no? Susceptible to SPS. Ibig sabihin yung SPS, no, yung SPS, it inhibits, okay, inhibits the growth, okay, of Peptostreptococcus anaerobius. Okay? Tatandaan din natin dito, recall question to no? Yung Peptostreptococcus anaerobius and Veillonella, they are both associated with jaw, okay, injury, okay, or jaw infection. Yan. And pakitandaan na lang din about Veillonella, it has a characteristic red fluorescence when exposed to UV light. Okay? So careful tayo dito sa jaw injury na ito. Okay? So pag tinanong gram positive anaerobic cocci nagkokos ng jaw-related infection or jaw injury-related infection, so that would be Peptostreptococcus anaerobius. Pero pag gram negative yung tinatanong, then that would be Veillonella species. Okay? Now let us proceed to anaerobic gram positive bacilli non-spore formers, because for the spore former, isa lang naman you have your Clostridium species, and I think we already have discussed about them. Okay? So, differentiate natin sila. All of them are catalase negative except for Cutibacterium acnes. Okay? And all of them also are indole negative except for Cutibacterium acnes. Okay? So, let us start with Actinomyces species. So, Actinomyces are fungus-like, no, organism. No? This is a fungus-like organism. Bakit siya tinawag na fungus-like? Because it has because it is a branching, no? It is a branching filamentous organism. Okay? Branching filamentous organism. So, kung meron tayong most notable member dito, that would be Actinomyces israeli. Okay? So, tatandaan natin yung kanyang characteristic white cerebriform. White cerebriform. You have the white cerebriform. Okay? Or dinidescribe din ito guys na molar tooth colonies. Okay? Also associated din ito, no, yung actinomyces natin, no, sa pagkakaroon ng tinatawag na sulfur granules. Okay? However, no, gusto ko lang i-mention din na si actinomyces, pwede siyang mapagkamalhan na sinocardia because of course of their characteristic morphology. Sinocardia kasi is also a fungus-like organism. However, to differentiate them, take note, actinomyces are actinomyces are acid-fast negative. Nocardia is acid-fast positive. Okay? Next, we have Cutibacterium acnes. This is formerly known as Propionibacterium. Okay? Yung dating pangalan niya is Propionibacterium. So this organism is has a diphtheroid, okay, diphtheroid gram stain morphology. Okay? Diphtheroid gram stain morphology. So anong ibig sabihin nun? Ibig sabihin it is resembling, no, the morphology of Corynebacterium diphtheria. Ibig sabihin nagkakaroon din siya, guys, ng Chinese letter, okay, arrangement, no? Then this is a skin commensal, which is why nga this is also a common contaminant of blood cultures, 'di ba? This is a skin commensal, and usually it causes infection of yung mga yung mga implants, no, such as prosthetic joint or yung mga CSF shunts, okay? So tandaan na, this is a skin commensal, and this is associated with infection, associated with prosthetic joint, or the better way of saying it is infection, no? Infection is associated with prosthetic joint and other foreign objects such as yung CSF shunt. CSF shunt. Yan. Okay? Then we have Bifidobacterium. So take note, catalase and indole negative. It is pleomorphic also. Okay? Pleomorphic. Okay? With ends. Pleomorphic with ends that may look like a dog biscuit. And lastly, we have Eubacterium, also catalase and indole negative, another pleomorphic organism, no? Pleomorphic organism, and is commonly associated with mouth and colon. Okay? Mouth and colon infections. Okay? And we now go to the last group for our anaerobic organism. We have our anaerobic gram-negative bacilli. Okay, so first, let's differentiate them based on their resistance, no, or susceptibility to vancomycin, kanamycin, and colistin. Okay, so all of them are resistant to vancomycin, okay, except for Porphyromonas. All of them are resistant to kanamycin, okay, except for Fusobacterium. And resistant to colistin or Bacteroides and Porphyromonas. However, Fusobacterium is sensitive to it. Prevotella, however, may be resistant or susceptible. All of them, or both Fusobacterium and Porphyromonas, are positive for indole. The other two are negative. And only Bacteroides fragilis is catalase positive. Okay? Now, the other three, except for Bacteroides fragilis, are fluorescent organisms, so no fluorescence for Bacteroides. Your Fusobacterium species usually has a chartreuse, okay, fluorescence, and brick red fluorescence for Porphyromonas, and red fluorescence for Prevotella. Okay? Now, take note of Bacteroides fragilis being the most common anaerobic pathogen. Okay, most common anaerobic pathogen. And take note of its 20% bile requirement. Okay, 20% bile requirement. Which is why, no, nag-e-exhibit siya ng black colonies, okay? Black colonies on BBE, no? BBE is bile Bacteroides bile esculin agar. Okay? Bacteroides bile esculin agar. So bakit siya nagkakaroon ng black colonies? Because Bacteroides is positive for bile esculin hydrolysis. Okay? So it is positive for bile esculin hydrolysis. It grows on 20% bile because it is bile resistant. Okay? Now, next we have Fusobacterium species. So dito meron tayong dalawa na i-differentiate natin. Of course, we have Fusobacterium nucleatum and Fusobacterium necrophorum. So Fusobacterium nucleatum and the other one is Fusobacterium necrophorum. So for Fusobacterium nucleatum, it is a fusiform, okay, or spindle shape, okay, fusiform spindle shaped organism, okay, whereas your necrophorum, okay, usually exhibit opalescent, okay, opalescent colonies with rainbow sheen. Okay? Opalescent colonies with the rainbow sheen. Yung colonies naman ng Fusobacterium nucleatum, take note yung breadcrumb. Okay? Breadcrumbs colonies niya. Okay? So, itong dalawa, no, pwede natin silang ma-differentiate with the use of lipase. Take note, ma-differentiate natin yung dalawa with the use of lipase. Okay? So, Fusobacterium nucleatum is lipase negative, whereas Fusobacterium necrophorum is lipase positive. So take note, yung blue na yan, that is for lipase. Okay? Next, let us proceed to Porphyromonas asaccharolithica. So it has brick red fluorescence when exposed to UV light. However, yung colonies niya, colonies appear, okay, brown to black on natural light. Okay, on natural light. So actually both Porphyromonas asaccharolithica and Provitella melanogenica, both of them are pigmented organism. Kasi pareho talaga silang merong black pigment, no? Pareho silang may black pigment. Okay? Take note also that the indole negative result for your Prevotella. Okay? Na pwedeng gamitin to differentiate it from Porphyromonas. No? Indole negative siya, Porphyromonas is positive. Both of them have black pigments. Both of them will have red fluorescence under UV light. Okay? So, today guys, usually, no, most of the laboratories, in order for them to be able to identify anaerobes, no, most laboratories are making use of ID of anaerobes. Okay? So most organisms, most laboratories are either using a kit, no? A kit, example of it is the Rapid ANA, no? The Rapid ANA kit, or pwede din yung Malditoff. Okay? Okay, so now let us proceed to spirochetes. Okay, so meron tayo ditong tatlo. We have Treponema, Borrelia, and Leptospira. So, first we have Treponema. So dito, pinaka-importante si Treponema pallidum, subspecies pallidum. Okay? So this is the agent of syphilis, of course, which is otherwise known as the great pox, the evil pox, or French or Italian disease. So this is a venereal disease, therefore it is transmitted through sexual contact. However, it can also be transmitted through blood transmission and can be transplacental. So technically, Treponema only have 3 stages, the primary, secondary, and tertiary. However, no, usually before the patient proceeds to tertiary syphilis, merong isang period wherein the patient will not exhibit any symptoms but remains seropositive. And that is the latent syphilis stage. No? So this is asymptomatic, okay, asymptomatic, however, seropositive. Yeah. Okay, so for primary syphilis, usually, no, this is characterized by having an ulcer, no, which is painless, no, tinatawag siya na hard chancre, which could last for 1 to 8 weeks. Okay? Then for the secondary syphilis, the characteristic chancre, the characteristic ulceration rather, is termed as condylomata lata. Okay? And for your tertiary syphilis, that would be gumas, no? Gumas, or tinatawag din siyang gumatas, no? Which is usually present sa skin or even sa bone. And of course, no, for the tertiary syphilis, this is now associated with cardiovascular, no? Cardiovascular and neurological involvement. Neurological involvement. I'll just rewrite. Okay, cardiovascular and neurological involvement. Okay? So that's why no yung neurosyphilis na tinatawag no that is already in the tertiary stage. Commonly no in the tertiary stage. However, we have other Treponema pallidum subspecies. We have Treponema pallidum perteniu, which is the causative agent of Yos. Okay? Treponema pallidum caratium, which is the causative agent of Pinta, and Treponema pallidum endemicum, the causative agent of Behel. Okay? So Yos and Penta can be transmitted through direct contact with lesions. Okay, direct contact with lesions, whereas Behel can be transmitted through sharing, okay, of utensils. Okay, next we have Borrelia. Okay, so dito meron tayong Borrelia burgdorferi and Borrelia recurrentes. So Borrelia burgdorferi is the causative agent of Lyme disease, which is a vector-borne disease, not transmitted by Exodus tick. Okay, Exodus tick. So similar to Treponema, no, or syphilis, meron din siyang tatlong stages. First is the erythema chronicum migrans, no, that is the reddening that is usually present in the site of the bite of the Exodus tick. Then for the second stage, it involves neurological manifestations and arthritis. And for the third stage, we have chronic arthritis and acrodermatitis chronica atropicans. Okay? So usually guys, no, Borrelia burgdorferi infection is nag-o-occur siya together with Babesia microti, no? Babesia microti infection. Okay? Dahil sa fact no na pareho sila ng vector. So pwede guys na may co-infection, no? It could exist as a co-infection with Babesia microti. Okay? Then the causative agent naman of relapsing fever is Borrelia recurrentes, and the vector is Pediculus humanus or the human louse. Okay? Then lastly, we have the Leptospira. So dito meron tayong dalawa, Leptospira interrogans and Leptospira biflexa. However, Leptospira biflexa is non-pathogenic. So Leptospira interrogans is the causative agent of leptospirosis and Wales disease. The Wales disease is the severe form of leptospirosis. Now take note of the specimen that needs to be collected depending on the time no or the duration of the infection. So usually on the first week of the infection, the organism can be isolated from blood or CSF. However, in the second week of the infection, the organism can be isolated from the urine. Okay? So it has a characteristic corkscrew motility. And take note guys, recall question. Okay? Media, no? Media for Leptospira, we have Fletcher's medium. Okay? Fletcher's medium. Also, I would like you to take note of the gold standard when it comes to the gold standard when it comes to the serological diagnosis of leptospirosis. That is microscopic agglutination test. Okay, microscopic agglutination test. Okay? So this is the gold standard for serological diagnosis. Okay? Gold standard for serological diagnosis. Okay? So yun po no yung ating mga spirochetes. So yung spirochetes natin also take note natin parate that they are difficult to stain no with the use of gram stain. So oftentimes they can be observed no if we want direct examination. So usually we require a dark field microscope for that. Okay, dark field microscope. Kasi nga hindi sila easily na-stain no yung spirochetes. Okay? So, now let us proceed no sa mga organisms that are usually virus-like. No, virus-like sila because they are obligate intracellular organisms. So that means kailangan nilang mag-thrive or they only thrive inside the cell. Okay? So this includes Rickettsia, Ehrlichia, Coxiella, Orensia, and Bartonella in the case of the Rickettsia species. Then later on, we also have Chlamydia and Mycoplasma. Okay, so these organisms are non-culturable with the use of our usual artificial media because of the fact again that they are obligate intracellular organisms. And so medyo katulad sila ng virus. However, in contrast to our viruses, yung mga Rickettsia, Mycoplasma, and Chlamydia, they contain both DNA and RNA. Kasi yung mga viruses, pwede lang silang magkaroon ng DNA or RNA lang. Okay? So, Rickettsia is of course a gram-negative organism and they are usually arthropod-borne. However, no, I would like to point that out later na meron tayong isang uri ng Rickettsia that is airborne. Okay? Now, for the laboratory diagnosis, we could make use of special stains such as Jimenez, Machiavello, Castaneda, and Gemsa. Okay? For the culture, again, hindi sila nakukulture sa artificial medium natin that are agar-based. No? So, it could be cultured on embryonated egg or pwedeng cell culture. Now, for serological diagnosis, we could make use of indirect immunofluorescent assay and the positive result should possess a titer of 1:64. Okay? Or pwede din natin gamitin yung Weyl Felix test, no? Which is a direct agglutination test based on the ability of Rickettsia no to cross-react or rickettsial produce antibodies to cross-react with certain proteus antigens. No, so yung ginagamit natin dito are OX2, OX19, and OXK. So take note lang natin that OX2 and 19 are derived from Proteus vulgaris, okay? Whereas OXK is derived from Proteus mirabilis. Okay? This is derived from Proteus mirabilis. Okay? So again, yung Weyl Felix test, this is a direct agglutination via cross-reaction kasi yung antibodies na napoproduce natin against certain rickettsial species can cross-react with Proteus antigens. And it could aid the identification based kung kaninong antigen siya nag-react, whether OX2, OX19, or OXK. Okay, so we have there the Weyl Felix result interpretation. So for epidemic typhus, it is positive for OX2 and OX19, however negative for OXK. Actually guys, makikita mo dito that the moment na you had a positive result for OX2 and 19, magne-negative ka talaga sa OXK. And vice versa, kung nag-positive ka sa OXK, negative ka sa 2 and 19. However, meron din tayong mga rickettsial species that do not cross-react, such as the Q fever and the rickettsial pox. Okay? So for the epidemic typhus, the causative agent here, the causative agent of epidemic typhus is Rickettsia prowazekii. Okay? Prowazekii. And dito gusto kong tandaan din ninyo yung tinatawag natin na Brill-Zinsser disease. Okay? The Brill-Zinsser disease, which is actually a reactivation only. This is a reactivation, okay, of the latent infection of Rickettsia prowazekii. Again, the Brill-Zinsser disease is a reactivation of latent infection caused by Rickettsia prowazekii. Then we have murine typhus, which is caused by Rickettsia typhi. Okay? Then we have the spotted fever group. Dito pinakatatandaan natin si Rickettsia rickettsii. Okay? Or Rickettsia. Yan. Okay? So this is the causative agent of Rocky Mountain spotted fever. And take note natin, this is the worst, okay, infection. Okay? So the worst form of infection that is caused by rickettsial species is the Rocky Mountain spotted fever, which is caused by Rickettsia. Okay? Then for scrub typhus, this is caused by Rickettsia chuchugamushi. Okay? So take note, siya lang yung nagpa-positive kay OXK. Then the Q fever, which is caused by Coxiella, okay? Coxiella burnetii. And ito guys, yung gusto kong tandaan nyo, that can be airborne. Okay? Kasi usually they are arthropod-borne, pero may isa tayo na pwedeng airborne, and that is Coxiella burnetii. And lastly, we have rickettsial pox, which is caused by Rickettsia acari. Okay, so yan po. No, so pakitandaan no yung kanilang Will-Felix reaction. Okay? Then let us proceed to chlamydia and mycoplasma. So let us start with chlamydia first. So katulad ng Rickettsia, it is also an obligate intracellular gram-negative organism that contains both DNA and RNA. So chlamydia has a special affinity to columnar cells, and considering na medyo mahirap siyang i-culture, no, pwede na nating gawing diagnostic test yung staining with the use of GEMSA stain. And maghahanap tayo ng characteristic inclusion body of chlamydia. So there are two types no of the inclusion bodies. The other one is the infectious, metabolically inactive form that is able to survive outside the cell. Again, infectious, metabolically active form that is able to survive, metabolically inactive form that is able to survive outside the cell, and that is your elementary bodies. Okay, elementary bodies. And the other one is the replicating form. And since it is replicating, of course, it has to be metabolically active. And again, they can only replicate inside, no, inside the cell. So they are not able to survive outside the cells. So that is reticulate bodies. Okay? Reticulate bodies, no? So ako guys, yung tinatandaan ko dito, medyo ano lang siya, pero tinatandaan ko elementary, infectious, tapos reticulate, replicating. Okay? So there are 3 chlamydia species. We have Chlamydia trachomatis, Chlamydia psittaci, and Chlamydia pneumoniae. So Chlamydia trachomatis is the trick agent. Trick means trachoma and inclusion conjunctivitis. Okay? Trick means trachoma and inclusion conjunctivitis. Okay? Also, this is the causative agent of lymphogranuloma venereum, specifically yung kanyang L1, L2, and L3 subtypes. Dito siya. L1, L2, and L3 subtypes. Yan yung nagkukos ng lymphogranuloma venereum. And for the trachoma naman, usually that is caused by the A, the B, the BA, and the C subtypes. Whereas the conjunctivitis is oftentimes caused by subtypes D, 2K. Okay? Now, we have a skin test no for the detection of lymphogranuloma venereum that is called as the Frey test. Okay? So this is the most common cause of non-gonococcal urethritis. It is sensitive to sulfonamides and also associated with Reiter syndrome. Then we have Chlamydia psittaci, which is the causative agent of psittacosis or ornithosis, or otherwise known as the parrot fever. Guys, take note that the mode of transmission is inhalation, thus no manifestation is pneumonia. Okay, it has non-glycogen inclusion bodies, and if trachomatis is sensitive to sulfonamides, psittaci is resistant to sulfonamides. Okay, so trachomatis is sensitive to sulfonamides, psittaci is resistant to sulfonamides. And the third chlamydia species is Chlamydia pneumoniae, no, or tinawag din siya na TWAR. Okay? TWAR because of the first two isolates of Chlamydia pneumoniae. The first two isolates are TW183 and the other one is AR39. Okay, so these are the first two isolates. No, so it has human-to-human transmission, also associated with Guillain-Barré syndrome. It is pear-shaped, large, no, periplasmic space and round EB. Okay, and in order for us to grow this organism, okay, kailangan natin ng cell lines din because again, no, it is an obligate intracellular organism. Okay? So take note that still no, for us to be able still no, the gold standard for chlamydia, okay, is cell culture. Okay? So pwede tayong gumamit ng makoy, no, or buffalo green monkey cells, no, for us to grow them. Of course, no, we could also directly visualize Chlamydia trachomatis elementary bodies in smears of clinical specimens with the use of direct fluorescent antibody tests. Now, we could also make use of nucleic acid hybridization tests, especially if the specimen is coming from urogenital and conjunctival specimens. No, this is still for Chlamydia trachomatis. But if the specimens are coming is an endocervical swab or a male urethral swab or pwede din yung urine samples, so usually we are making use of nucleic acid amplification tests. Okay? So that would be all no for chlamydia. And finally, okay, we have the Mycoplasma species. So Mycoplasma is the smallest free living organism known. Okay? Take note that Haemophilus ducreyi is the smallest pathogenic organism. Yon. The smallest free living organism known is Mycoplasma. So this one lacks a cell wall. That's why common recall question dito, yung tao daw merong pneumonia. Nung binigyan daw ng penicillin, hindi nag-respond. Bakit? Because the organism do not have a cell wall. So instead, it has sterols, pleomorphic siya, and it's non-motile. It can grow in artificial media, unlike the first two, the rickettsia and the chlamydia. This one can grow in artificial media. It is penicillin resistant and it has fried egg mulberry colonies. Now for the culture, pwedeng E. agar, pwedeng Shepard's A7B agar. Okay? So for the Mycoplasma species, of course, no, si Mycoplasma pneumoniae, common recall question. It is an Eton agent and a causative agent also of primary atypical pneumonia or tinatawag din ito na walking pneumonia. Okay? And this is associated, of course, with the production of cold agglutinins. That's why associated ito sa tinatawag natin na cold agglutinin disease. Okay? Cold agglutinin disease. Okay? Then we have Mycoplasma hominis. It has large fried egg colonies. Okay? So usually, no, fried egg yung colonies nila. Okay? Associated with post-abortal fever, postpartum fever, and pelvic inflammatory disease. Okay? Pelvic inflammatory disease. And lastly, okay, we have ureaplasma, urealyticum, also known as genital mycoplasma. It has a tiny fried egg colonies, urease positive, and the causative agent of non-gonococcal urethritis. Okay? So yan po no yung ating mga members no ng mycoplasma. Okay? And I think we are down to our last, okay, a group of organisms no for bacteriology. We have the Mycobacteria species, no. So first, take note muna natin no yung flow chart no of specimen processing to isolate Mycobacteria. Guys, I want you to take note of this, okay? Recall question din ito, yung tamang pagkakasunod-sunod daw ng processing ng specimen no for the isolation of Mycobacteria. So it is liquefaction or tinatawag din ito guys na digestion with the use of NALC, no? NALC or N-acetyl L-cysteine, followed by decontamination with the use of sodium hydroxide, followed by neutralization, and lastly centrifugation at 3,000 G, no? 3,000 G for better yield, no? For high yield. Okay? So usually, no, ginagawa natin itong apat na stage na ito or apat na steps na ito if the specimen is a non-sterile specimen. Okay? But if it is sterile, you do not have to do decontamination anymore, di ba? Now, the organism, no, can be grown on liquid media. It can also be grown on solid media. Okay? So commonly for the solid media, we are using the egg-based medium. We are using the agar-based medium, or we are using the egg-based rather, no? Kasi Lowenstein-Jensen yung pinaka-common na ginagamit. So Lowenstein-Jensen is egg-based, okay? But pwede din tayong gumamit ng agar-based, which is Middlebrook, no? Because it is agar-based. Okay? So incubate natin siya, no, at 37 degrees Celsius for more than 8 weeks, no? It could reach, no, up to 8 weeks, okay, for a mycobacteria, no? Because meron tayong mga slow growers na tinatawag, okay? And if growth occurs, i-confirm natin gamit ng acid-fast staining. Okay? Pwede din natin siyang i-grow sa liquid medium. Now, ano ba itong MGIT? No? Gusto ko lang siyang i-expound. MGIT is, yung ibig sabihin nito is mycobacteria, okay, growth indicator tube. Okay? Mycobacteria growth indicator tube. So usually, no, pwede din itong i-incorporate or pwede siyang gamitin for us to be able to detect drug-resistant strains. Kasi usually daw, no, dito sa MGIT, na naglalagay, when the organism grows, no, in the MGIT, that means that organism is a drug-resistant organism. Okay? So meron tayo ditong fluorescent indicator that once it fluoresces, ibig sabihin nag-grow yung organism. Okay? So that's MGIT, no? So it is capable of detecting drug resistant strains. Yeah. Okay? So for DSSM, okay, so of course, no, the specimen must be collected in a sterile, completely labeled specimen cup. The volume of the sputum should be at least 1 teaspoon, so that is approximately 5 to 10 ml. Now dati pa yung 3 specimen. Ngayon, 2 na lang yung specimen requirement, no, for DSSM, no, 2 specimens na lang. So the first specimen is during your first consultation. So that could be a spot specimen. And the second specimen should be provided 1 hour to 3 days after the first specimen. Okay? So 1 hour to 3 days after the first specimen. So the method, pwede tayong gumamit ng Ziehl-Neelsen or fluorescence microscopy with the use of uramine-rhodamine stain. Okay? Uramine-rhodamine stain. Yung Kinyon, usually ginagamit yan for tissue specimens, no? Kasi yan yung tinatawag natin na tissue AFS. Okay? So in order for you to be diagnosed, okay, na positive, you have to at least, no, give a positive result for at least one of the specimens. Okay? Now, usually ano yung setup, no, for the detection or the identification of Mycobacteria species. So yung unang ginagawa technically is, no, eco-culture, 'di ba, no? So eco-culture natin yung specimen, no? So it could be in a liquid media or it could be in a solid media. Okay? So usually, no, for una eco-culture natin, no? Eco-culture, pwede natin siyang i-culture sa solid, no? Pwede siyang i-culture sa solid media. Okay? So pag solid, either agar-based 'yon, no? Agar-based. Okay? Or pwede siyang egg-based, no? So pwede siyang egg-based. So usually, no, sino yung mga example ng agar-based? Yung pinakakilala dito si Middlebrook. Okay? Si Middlebrook, 7H10 and 7H11. Then we also have Mitchison's. Okay, Mitchison's medium. Then we have the Du Bois oleic acid. Okay, oleic acid albumin medium. And okay, now for the egg base, which is the common, no, common one. So nandito yung ating LJ bottles, no, the Lowenstein, no, Lowenstein, Jensen. Okay, medium. Then we have Petragnani. Then we have the American Thoracic Society medium. Okay, the American Thoracic Society, okay, medium. And lastly, we have the Dorset, okay, medium. Now, pwede din siya guys i-culture gamit ng liquid media or yung broth natin. So usually yung broth ginagamit ito for rapid, no? For rapid identification, no? Kasi mas mabilis siyang mag-grow gamit ng liquid media. Okay? So example ng mga liquid media natin, no? Of course, usually ito yung ginagamit natin for automation, no? So nandito yung ating Bactec. Okay? Bactec 12B. Okay? Nandito din yung SeptiCheck. Okay, the SeptiCheck AFB, and we have the Middlebrook. Okay, Middlebrook 7H9 and 7H12. Okay, so yung 7H10 and 7H11, they are solid base media. So usually, no, isa din sa possible, no, na characteristic na pwede nating ma-exhibit gamit ng liquid media is yung cording ng Mycobacterius tuberculosis. Okay? So before we go, no, So I was mentioning, no, paano daw yung pagkakasunod-sunod ng identification, no, ng mycobacteria. So usually magsa-start tayo sa culture, and through this, no, i-differentiate natin sa whether, no, it is a rapid, no, rapid or slow grower. So yan. Ito na lang. So usually i-differentiate whether it is a rapid, no? So first, differentiate whether it is a rapid or slow grower. Then after that, no, the second step, usually chine-check yung temperature preference. Okay? The temperature preference. Or commonly colony, yung colony pigmentation. Or colony pigmentation, which is one, no, which is one of the basis, no, of the classification done by Runions, no, yung Runions classification of Mycobacterium other than tuberculosis. 'Di ba? Meron tayo dong photochromogens, non-photochromogens, and scotochromogens. Okay? Then, and lastly, of course, no, i-differentiate na natin siya gamit ng mga biochemical tests. Okay? And usually, no, yung pag-identify natin with the use of biochemical test, this may take up to 2 weeks, no? Pwede siyang mag-last up to 2 weeks, no, to ultimately identify, no, the Mycobacterium species. Okay? Present. Okay? So of course, no, yung pinaka-importante dito na dapat alam na alam natin, okay, would be, okay, Mycobacterium tuberculosis. Siya talaga, no, yung pinaka tatandaan natin dito. So ano ba yung mga bagay-bagay, no, na tatandaan natin about the characteristic of Mycobacterium tuberculosis? Okay? So it is a slow grower, no? Mycobacterium tuberculosis is a slow grower na pwede siyang umabot ng 2 to 6 weeks, no, bago siya tumubo. Okay? And the organism will usually grow flat, dry, okay, white and wrinkled colonies. Okay? On solid media. Okay? On solid media. Now, so that gives us the idea that Mycobacterium tuberculosis, considering that it is white, no, it is a non-chromogenic or non-pigmented organism. Okay, non-chromogenic or non-pigmented organism. So usually yung kanyang preferred by the way, on solid media, no, it is flat, dry, right, white or wrinkled colonies, and it could exhibit its cording, no, cording, okay, in a broth or liquid media. Okay? And take note na yung kanyang preference, yung temperature preference niya is 37 degrees Celsius. Okay? So that would be all for Mycobacterium tuberculosis. Now, other than, of course, no, the culture, no, or the acid-fast staining, the culture, meron din tayong mga other diagnostic tests to be able to identify whether the person has tuberculosis. So, I mentioned here dalawa. We have the TST, no, the TB skin test, or tinatawag din natin ito na PPD, no, kasi gumagamit tayo dito ng purified protein derivative. So this is a skin test, no, a skin test. So, however, guys, no, this is not that specific when it comes to the diagnosis of tuberculosis. Why? It cross-react with BCG vaccine and it cross-react with other atypical mycobacteria. Therefore, pag nag-positive tayo dito, no, pag nag-positive ka sa TB skin test, there are 4 possible causes of that. One is, pwedeng meron kang active TB. Okay? Number 2, pwede kang merong latent TB. Okay? Number 3, pwede kang merong infection, no? Infection with non-tuberculosis mycobacteria. Okay, infection with non-tuberculosis mycobacteria. And number 4, pwedeng kakabaksin mo lang, no, vaccination with Bacillus calmette-guerin. Okay, so hindi siya ganun ka-specific because again, pag nag-positive tayo, no, sa TSST or the TB skin test or purified protein derivative, pwede yung interpretation, it could be active TB, it could be latent TB, pwede na yung infection mo is because of other non-tuberculosis mycobacteria, or it could be because of your vaccination with Bacillus calmette-guerin. Another thing, remember guys na yung PPD test is classified as a delayed hypersensitivity reaction, di ba? This is a delayed hypersensitivity reaction. So that means medyo matagal yung result, no? Pwede siyang umabot ng 72 hours. So, in contrast to IGRA, which is referred to as which means interferon gamma. Sorry. Mali yung spelling. Okay? IGRA, no, na referred to as interferon gamma release assay. Isa sa mga advantage nito, mabilis siya, no? The result can be released, no, within the day. It does not cross-react with BCG and it does not cross-react with other atypical mycobacteria. That's why pag nag-positive ka sa IGRA, automatically meron ka talagang latent tuberculosis. Okay? So take note ha, recall question yung IGRA na yan. Okay? Parang may isang tanong na positive siya sa IGRA, negative siya sa TB skin test, ano daw yung ibig sabihin? Parang ganun. Ay baliktad. Positive pala siya sa TB skin test pero negative siya sa IGRA. So it could be because of the vaccination with BCG. Okay? Tapos may isang tanong din na ano daw yung advantage ng IGRA over TSST. Tapos yung nandun daw sa choices is the rapid results. Okay? So yan. Now, I already have mentioned about the cultural characteristics of Mycobacteria, no? The characteristics of its colonies on solid media and the cording no, when it comes to liquid media. I want you also here to take note of the key biochemicals when it comes to Mycobacterium tuberculosis. Tatlo lang naman yun guys no? So bigay ko na sa inyo yung ibang mga positive result dito. But I want you to take note here of Mycobacterium tuberculosis talaga, yung kanyang key biochemical reaction. So it is positive for niacin, positive for nitrate, and negative for 68 degrees Celsius catalase. Yan yung key biochemicals, no, for mycobacteria. Now let us proceed to mycobacteria other than tuberculosis. No, this is the Runions. No, this is the Runions classification. Okay, so dito naka-classify siya based on whether it is a slow or rapid grower. So ito yung mga rapid growers natin. Then 'di ba after classification whether it is a slow or rapid grower, ika-classify mo siya based on its pigmentation. Okay? So for the group 1, we have the photochromogen. So yung mga photochromogen, they develop yellow pigment when exposed to constant light source, pero non-pigmented sila in the dark. Okay? So sino yung mga members dito? We have Mycobacterium marinum, Mycobacterium asiaticum, Simiae, and Kansasii. Guys, take note, Mycobacterium marinum is usually associated with wound infection exposed no sa mga fish tanks, mga ganon. That's why this is associated no with fish tank, or this is the causative agent of fish tank granuloma. Yan. Then si Mycobacterium kansasii, no, this is referred to as the yellow, no, the yellow bacillus. And this causes a chronic pulmonary disease, no, that resembles TB, no? A chronic pulmonary disease that resembles TB. Paano ba? Chronic pulmonary disease that is TB-like. Yan. Okay? Then, we have the group 2. The group 2 are the scotochromogens. No? Ano naman yung characteristic ng mga scotochromogens? They are producing yellow to orange pigment in the dark, and the pigmentation intensifies to orange or red when exposed to constant light for 2 weeks. Okay? So, sino yung mga members dito? We have Scrofolaceum, Sinope, Gordonei, Flavescens, Thermoresistible, and Zulgay. I want you to take note of Gordonei, recall question. Of course, no? Alam natin to. This is the top water bacillus. Okay? And for the group 3, we have the non-photochromogens. Nandito si Malmoense, Avium intracellulare, Terrae triviale, and Haemophilum. Okay? So, I want you to take note here first, no, of Terrae triviale. Ito yung tinatawag natin na radish. Okay? The radish bacillus. Okay? And of course, no, I want you to take note of Mycobacterium avium intracellulare. Okay? So ano ba yung mga characteristics na tatandaan natin sa kanya? Okay? So ito. Mycobacterium avium intracellulare. So of course, itong MAC complex na ito, intracellulare, this is composed no, of 2 organisms, the Mycobacterium avium and the Mycobacterium intracellulare. So usually, no, ito yung causative agent ng tuberculosis, no, the causative agent of tuberculosis in immunocompromised patients. Okay? In immunocompromised patients, no, such as yung may mga AIDS. Okay? Patients. And of course, nagkukos din siya guys ng infection sa mga immunocompetent, no? Pwede din siyang mag-cause ng infection sa immunocompetent individuals. And usually, no, there are 3 forms of infections, no, sa mga immunocompetent individuals. No. O sabihin na lang natin immunocompetent infections, no? So, the first form is a upper lobe cavitary disease. Okay, an upper lobe cavitary disease that is also resembling tuberculosis, no? So lagay na lang natin TB-like no, that is seen in heavy smokers. Yeah. Okay? Next is yung tinatawag na Lady Windermere syndrome. Okay, the Lady Windermere syndrome. Okay, which is characterized by having a weak cough, no? Okay, characterized by having weak cough. Okay, that is often seen in elderly, no? In elderly women. Okay, and the third form of the disease is a hypersensitivity reaction. Okay, hypersensitivity reaction. Okay, after exposure to hot tub contaminated with the organism. So kumbaga, no, nagkaroon ka ng hypersensitivity reaction with the Mycobacterium avium intracellulare whenever it is present, no, sa hot tub, no, na kung saan ka na-expose. Okay? So yan yung mga tatandaan natin kay Mycobacterium avium intracellulare. Now ito guys, no, because this came out of my exam and I actually answered it wrong, no, kasi yung sagot ko doon is Mycobacterium tuberculosis, no? Pero ito siya. Yung Mycobacterium, medyo na-confuse kasi ako sa tanong because I know for a fact that Mycobacterium avium intracellulare is a non-photochromogen. So, technically, it does not have any pigment. However, in that question, no, the organism daw is a slow grower, no? The organism is a slow grower with smooth, no, smooth yellow to orange colonies, parang ganon. So, I do not know ba kung bakit pa ako sumagot ng Mycobacterium tuberculosis, alam ko naman na rough yung MTB na colonies. Pero ito siya guys. Si Mycobacterium avium intracellulare, no, it can become yellow to orange with age. So yes, the colonies are non-pigmented. However, habang tumatagal, it could turn to yellow or orange. So take note recall question ko to sa exam ko. Slow growing siya. Okay? Slow growing. Actually, LJ medium yon, no? Tinanim daw siya sa egg-based medium. So slow growing, then it's initially non-pigmented but become yellow, no? It become yellow to orange with age. And of course, it also has smooth colonies. Okay? So, take note of this. Okay, baka lumabas ulit sa exam. Yeah. Okay, so that is MAC, no? Mycobacterium avium intracellulare complex. Yeah. And we now go to the last group. We have the rapid growers. So nandito guys si Fortuitum salonae. Kung meron tayong tatandaan, no, sa kanilang dalawa, this is the only group of organism that is positive for aryl sulfatase test. No? So sila yung positive sa aryl sulfatase test. Okay? Then si Smegmatis, of course, ito naman yung tinatawag na urine, no? The urine acid fast bacillus. And take note that Mycobacterium flay has a CO2 requirement. So that would mean na si Mycobacterium flay is a capnophilic mycobacteria. Okay? So I think no that ends our topic on bacteriology. Okay? Maraming salamat guys.